Now we'll see what are the proteins' tertiary and quaternary structure. So these are the two most advanced structures of proteins. The tertiary structure is the three-dimensional shape of the proteins that is formed due to interactions and repulsions of amino acid residues. So we will see more into like what type of interactions will make this tertiary structure. So you see a ribbon model of a tertiary structure of a protein. So now you see that this is a three-dimensional structure because there are multiple interactions that happens in between amino acid residues of a given secondary structure. So what are the interactions that makes these tertiary structures? So this is one most important part in this lecture series to identify what are the interactions that make these three-dimensional structures of proteins. So as these are globular proteins or as these are three-dimensional structures of proteins, there should be some stabilizing interactions to keep the shape of these proteins. So these stabilizing interactions can be identified or interpreted as different using different types of interactions. The first one is called hydrophobic interactions. Now we know what are these hydrophobic interactions. So these are occurred due to the interactions from non-polar residues. So these are interactions between two amino acids that have non-polar R groups. For example, you see here a side chain or an R group of a phenylalanine, which contains the aromatic group, and leucine, which contains this isopropyl group at the end of the R group. So this is an example for um, hydrophobic interactions. So when there are such nonpolar R groups in two amino acids, these interactions are hydrophobic interactions. So when there are such amino acids present in a uh, tertiary structure, so that can make a nonpolar center at the interior of the protein. Um, So always these nonpolar uh, groups will pushed inside the protein because like these proteins exist in our systems where most of the parts are aqueous medium. So these nonpolar groups doesn't like to expose to water. So as a result, always these nonpolar groups will just push inside the protein molecule, and polar groups will come outside and non-polar groups will always stay inside of the protein. So this is one type of interaction, which is hydrophobic interaction. The second type of interaction is the hydrophilic interactions. You know what type of groups will make these hydrophilic interactions. So obviously, those have to be polar amino acid residues. Those are the amino acids that contains polar R groups. For example, serine amino acid contains the CH2OH group. When there are polar groups like hydroxyl, they like to make interactions with water, right? So simply those are hydrogen bonds. So oxygen atom and both oxygen and hydrogen can make hydrogen bonds with water molecules. Um, so these type of amino acids always will like to expose to outside aqueous environment because they like to dissolve in water. And these type of amino acids will pull to the outer surface of most proteins. So those are hydrophilic interactions. And another interaction that makes these tertiary structures are called salt bridges. So as the name implies, the salt bridges are the ionic interactions, which means the salts are ionic compounds, right? Which contains a negative ion and a positive ion. So the interaction happens in between these negative and positive charges. If you see a group or an R group that contains a negative charge, like in carboxylate groups, and an R group that contains a positively ion charge, together they can make these salt bridges. So these are ionic interactions. Salt bridges are same as ionic interactions. So based on the amino acid category, so these interactions occur in between polar basic amino acids and polar acidic amino acids. So those are ionic interactions, positive and negative charged interactions. So that's the third type of interactions that will occur to make a tertiary structure of a protein. And the fourth one is one more major important bond called hydrogen bonds. So hydrogen bonds will form between a hydrogen of a polar R group and the oxygen or nitrogen of any other polar amino acids. So in order to make a hydrogen bond, the hydrogen atom connected to any other electronegative atom in a polar amino acid, and it has to be connected to either oxygen or nitrogen of another polar amino acid. So these are hydrogen bonds. Hydrogen bonds are also playing a major role to keep the tertiary structure in its shape. And another type of interaction is called disulfide bonds. So these disulfide bonds are made in between cysteine residues. So cysteine contains these thiol groups, SH groups, and a disulfide bond is a covalent bond, and these covalent bonds can be made by in between these cysteine to cysteine residues. So disulfide bonds are strong bonds because these are covalent bonds, and those are also playing a major role to keep this tertiary structure in its place by keeping these strong bonds. Okay, so we know now different types of interactions that keeps this tertiary structure in its place or in its shape. So we will summarize all of them here. So as I mentioned, the hydrogen bonds will help to keep the structure. So hydrogen bonds occur in between a hydrogen atom that connected to any electronegative atom such as oxygen or nitrogen, and it has to be connected to another electronegative atom, oxygen or nitrogen. So that is called a hydrogen bond. So hydrogen bonds will play a major role on salt bridges in between positive and negative charges, and disulfide bonds, the bonds in between two sulfur atoms, these are covalent bonds, and hydrophobic interactions in between these hydrophobic R groups, um, and hydrophilic interactions with water. Now you can see usually these hydrophobic interactions will happen inside the protein structure, where hydrophilic interactions are occurred outside of the or surface of the proteins. Usually, generally, the hydrophobic interactions or hydrophobic R groups will just pushed inside the protein. So all of these different types of interactions helps to keep the tertiary structure in its shape, and so the bond strength and bond stability is very important to keep them to keep their three-dimensional shape. Okay, now we will see what is the quaternary structure of a protein. And to describe the quaternary structure, I'm using this example, hemoglobin protein. So we know that how the tertiary structure is maintained. Now, the quaternary structure is just the combination of two or more polypeptide chains or subunits um, together. For example, this hemoglobin protein contains four subunits or four tertiary structures combined together to form this hemoglobin protein. Now, just to talk a little bit about hemoglobin protein. So this is a globular protein which has a shape of a sphere, and that helps to transport iron, transport oxygen in blood. So it consists of four polypeptide chains, two alpha chains with 141 amino acids. So these two alpha chains are made up of 141 amino acids and two beta chains with 146 amino acids. So the subunits are held together by the same stabilizing interactions in tertiary structures. So we talked about so many different types of interactions. So those are the ones that helps to stabilize these subunits together. Each subunit of hemoglobin is a globular protein with an embedded heme group. So heme group has this ion center that can bind the oxygen molecule. So this is an amazing creature by the nature itself, because this hemoglobin protein is one of the most important protein for our biological functions, right? Without hemoglobin, uh, we can't exist because oxygen is needed for any living being. So oxygen molecule is contained within this heme group, and the heme group is present in all of these four subunits in the hemoglobin protein. So, uh, so the heme group has the ability to trap the oxygen molecule inside. So that's how this hemoglobin protein is transporting oxygen in our blood. So this hemoglobin structure is an example for a quaternary structure. So now you can imagine how this structure primary structure is simply a sequence of amino acids linked by the peptide bonds. And the primary structure will organize to give a secondary structure by having these hydrogen bond interactions. So the secondary structure can be an alpha helix or beta plated sheets. And then the secondary structure will organize to make the tertiary structure by those different types of interactions. They will keep it or maintain the shape of the tertiary structure. So by combining multiple tertiary structures together, the quaternary structures are formed. So these are the most complex proteins. So this is how the protein structures are designed or organized, starting from a single amino acid.